Thank you. It's really a pleasure to be here with all of you today. I work with a biotechnology company called Decibel Therapeutics. We are focused on developing therapies for people with hearing and balance loss. We're a publicly traded company, so I need to let you know I may be making some forward-looking statements today. All right, so we're going to spend our time today focused on congenital hearing loss. We'll be talking about diagnosing hearing loss in infants and the role of genetics. You know, the hearing loss field, there's a really exciting story here over the last few decades where, you know, it used to be the case that it took us a number of years before we were able to diagnose children with hearing loss. We sort of needed to wait until parents noticed that their children were falling behind in speech language development. And through the work of a number of folks over the last few decades, including Carl White, we're now at the place in the US where around 98% of kids are getting screened for hearing loss using physiologic assessments before they leave the hospital. What we've found as we've taken on these efforts is that hearing loss, congenital hearing loss, is relatively common. So 1 in 500 newborns are going to be diagnosed with hearing loss. And as we've learned more about these children, we've also begun understanding that many of these cases of hearing loss are actually caused by genetic protein deficiencies. So I've highlighted a few here, and we'll talk more about it today, but one of them that we'll spend some time talking about is Otoferlin, which is on chromosome 2. If you've heard about any genes that might cause or might be related to hearing loss, you maybe have heard of GJB2 or connexin 26. This one is a common gene associated with hearing loss in kids. Others are genes like myo7A, for instance, it's involved in Usher syndrome. For the kids that we're seeing, most of these cases are non-syndromic hearing loss. And one of the things I wanted to point out is they're generally autosomal recessive. And this is not news or anything surprising to anyone with genetics training on the call, but I bring this up just to say, just from a perspective of the parent for a second, you know, most of the parents of these kids who are born with hearing loss have a typical hearing themselves. And so for them, getting this diagnosis very early in life is a lot to take in. There's a lot to learn and a lot of uncertainty during that process. And so one of the jobs of the clinician in the early phases of this diagnostic process is really helping the family learn, understand, and be able to accept this diagnosis. So in order to have this conversation today, I want to spend just a moment here talking about hearing, how it works, and some of the cells involved, because we'll talk a little bit with some more specificity about some of them. So the basics here I'll point out. If you look at this picture on the left-hand side, when we think about the ear, people oftentimes talk about it sort of in 3 different stages. So they'll talk about the outer ear. If you look at the outer ear, that's the pinna, and then your ear canal where you can kind of stick your finger in a bit, where you can get wax built up, things like that. That's the outer ear. The middle ear, if you follow that ear canal in this picture to where it's terminating here with the eardrum or the tympanic membrane, that's the beginning of the middle ear space, which is sort of this open air cavity here where you can see you have the eardrum connected to 3 bones. This is the space where, you know, sometimes kids get ear infections, for instance, and they may feel some pain. They may have some hearing loss associated with that. And that's because they have fluid built up in this space that's normally filled with air. And what's happening in those cases is you can impede the ability of the eardrum and these tiny bones in this middle ear space to actually transmit mechanical energy to the inner ear, which is over here on the right-hand side, also called the cochlea. So the cochlea is snail-shaped, and it's filled with fluid. And so if I step back for a second here, when you have sound out in the environment, these are pressure waves. They come in the ear, they go down the ear canal. They're going to be hitting against your eardrum, and just like a good drum, you'll get movement of the eardrum. That movement gets transmitted also to these tiny bones, and ultimately they're sitting there. These bones are pushing in on a fluid-filled space, so they are creating vibrations in that fluid. So they actually create these little tiny waves that are moving around in the inner ear and stimulating different sensory cells. There's a lot of complexity to talk about there, but I'd actually just want to simplify for a second and say that if you look deep inside that ear now, what you'll find in any given part of that ear is that there are a couple of types of sensory cells that are going to be responsible for detecting these vibrations that are being created by sounds and turning them into signals that can be sent on to the brain. So here are the two types of cells. You have this cell right here that I'm pointing to, the so-called inner hair cell. And then you have these cells over here, which are the so-called outer hair cells. So your inner hair cells are the primary transmitter of information from your ear to your brain. So when you have vibrations in the inner ear, they actually stimulate these little hair cells. These little hair cells then will signal to the auditory nerve, which is the eighth cranial nerve, and send messages on to the brain. You have 3,500 of these inner hair cells sort of spread out side by side across the length of your cochlea. And they're tuned like a piano is tuned. So in one region of the cochlea, they respond to low-pitched sounds, and to another region on the other side, they respond to high-pitched sounds. You see next to them, you have these little outer hair cells. So what do? Well, they don't send very much information to your brain. You have 3 times as many of them as you have of the inner hair cells. And what they end up doing is they actually amplify soft vibrations or soft sounds so that they can be detected by the inner hair cells. So they're a little amplifier that's built into your ear. They also help tune. They help tune the responses as well. As I mentioned, your ear is sort of structured like a piano, and it's a very exquisitely mechanically tuned organ. So that's the basic pathway. You have vibration, or you have pressure waves out here that are sound. They get turned into vibrations along the way to your inner ear, and then they ultimately will stimulate these little hair cells that'll send signals onto the brain. And so how do we assess hearing and the function of these cells in babies? Because that's what we're going to talk about today. Well, objective tests, of course, are quite key for us, because if you're thinking about a 1-month-old or a 2-month-old, it's going to be hard to do behavioral assessments for the child that age. So we use a couple of tests pretty frequently in the clinic. We also use these tests in newborn screening programs. So the one on the left-hand side here is called the auditory brainstem response. If you look at this baby in the center, they're having an auditory brainstem response being measured. So they have surface electrodes on the scalp, and then you have some earphones on the ear. You play some sound into the ear, and then you simply measure neural response to that sound with these surface electrodes. So if you see this gray line here where it has these little bumps, the first one that's labeled 1 is actually the response of the auditory nerve to sound. So that lets you know that the nerve is responding to the sound that you put into the baby's ears. If you follow those peaks up, that's neural activity at ascending levels in the auditory system. So it goes to the brainstem, and then you can follow it up to the midbrain with this particular measurement. That's assessing here as you look, the inner hair cell, again, signaling the auditory nerve. What about these other cell types, these outer hair cells? We can make a measurement called otoacoustic emissions. So we play sound into the ear. And because these cells will actually change their conformation to help this, provide this amplification in the inner ear, we can actually measure sound coming back out of their ear. So we put a tiny microphone in the ear canal, we can actually measure it coming out. And that's how we assess the status and the function of these cells. That's a test called otoacoustic emissions. Now, what's really important here, as I said, is that you can, with these types of tests in an infant, actually start understanding, which cell types might be affected when you're identifying a child who may have hearing loss. So here's an example of a child who does have hearing loss. So I showed you I pointed out the typical traces above. Here's an infant who has hearing loss. It's a 4-month-old. And you see that the clinician has played sound into the ear. They've tried to measure neural responses, but what you see in the red and blue is a flat line. So they played the sound as loud as they could. They could measure no response. That means the communication here, something in this pathway in the inner hair cell and the auditory nerve, there's a loss of information, basically, or loss of function. If you look over here on the right-hand side, what's kind of interesting, as you can see, is that the responses of the outer hair cells actually look good. So in this patient, they have they have loss of function here around the inner hair cells, but their outer hair cells are actually still functioning. Now, because at the end of the day, what matters is what information is being sent from your ear to your brain, this child still has profound hearing loss, even though some of their cells within their ear are actually functional. It turns out in this particular case I was showing you, this child has a subclass of sensory neural hearing loss called Auditory Neuropathy Spectrum Disorder. And that's why the title to my presentation said Auditory Neuropathy. It's because I wanted to spend a little bit of time talking with you about this sort of subclass of pediatric sensory neural hearing loss. The way that this is diagnosed is that essentially you look for this kind of discordance between the auditory brainstem response and the otoacoustic emissions. So usually for many types of hearing loss, the function of these sensory cells kind of change in concert. So many types of protein deficiencies and other insults to the ear kind of affect both of these cell types pretty similarly. So the test results kind of go in the same direction in many cases of hearing loss. In about 5 to 10% of cases, we see clinical presentation that looks like this, where one of the tests says profound hearing loss and the other test says, hey, we actually see pretty good function here still. Now, because what we're seeing here is essentially that there's some loss of function either of the inner hair cell itself, there might be a loss of the inner hair cell, the communication between the inner hair cell and the nerve, or maybe some dysfunction of the nerve itself that could produce these types of test results. It was back in the 90s when a group of clinicians and scientists named this auditory neuropathy. You know, the only way that they were actually able to make this diagnosis is that we had developed types of tools to actually assess cellular function across at least these two cellular populations. So prior to then, it wasn't that kids didn't have this type of hearing loss. It was just that we could never measure it because we didn't have this other test over here called otoacoustic emissions. But, you know, since then, so sort of over the last 30 years, we've started learning more about auditory neuropathy as a specific sort of subgroup of sensory neural hearing loss. And what we've seen is that there are a lot of different causes that can create this clinical presentation. So there are a number now of known genetic causes of auditory neuropathy. So these are specific protein deficiencies that affect either the inner hair cell, the synapse, or the nerve itself. There are a number of environmental and acquired factors. Also, autoimmune disease has been associated with this type of clinical presentation. And because there's sort of varied causes here and quite a bit of complexity with this clinical diagnosis, there are a number of opportunities here for the diagnostic process to actually be extended for this particular subgroup of patients. So let me tell you about it. So first off, there's some real challenges in trying to interpret some of the test results you get from patients like this. So in some kids, for instance, we might measure their ability, let's say, to detect that a sound is present. This is a pretty standard hearing test, as you play a tone and you ask if the child can hear it. They do kind of a head turn to show you that they hear something when they're very young. And what you might learn is that the child can detect that a sound is present. So that the visual example of that would be something like, you know, detect that you flip the lights on or something. So they can detect that a sound is present. But then when we test their ability, for instance, to understand a more complex signal, like be able to understand speech, for instance, the child's unable to do it. And that's because this synapse, while it might support something very simple, like being able to detect that a sound is present, it's insufficient to code a complex signal like speech or music, for instance. So anyway, so you might have a child who seems to detect things, but they can't understand anything. And so it sometimes takes more time diagnostically to, particularly in a young child, to be convinced of the diagnosis that you're making. Another challenge here is that there are comorbidities. So many of the children will have morbidities that you're also trying to take into account as you're getting the diagnosis. The the third here that I'll point out is that there are even some forms of this that are fluctuating. So there is a variant of children who have auditory neuropathy that is temperature sensitive. So their clinical presentation changes when their body temperature increases, 1 or 2 degrees. So in all here, what we see for these patients generally is it takes longer to get to diagnosis. It takes longer to be able to get to clear management decisions for many of these kids. All right, so once a management decision is made, there are a few different paths that many of these kids will go down, and many of them might do sort of a bit of all 3 of these different subgroups. So one is that the children may use some forms of assistive devices. So those could be hearing aids or frequency modulation systems. They might become candidates for cochlear implants. So cochlear implants are a neural prosthetic, and the basic idea here and sort of the way this works is that an array of electrodes is surgically implanted up near the auditory nerve. A child wears a microphone and signal processor outside of their head, and we collect auditory information from the environment, and then we use that information and code it to directly stimulate the auditory nerve and essentially bypass the ear to provide the brain some input and access to sound. So many kids with auditory neuropathy, in fact, will become cochlear implant candidates at some point. Additionally, so in addition to these sort of device-based management strategies, there are many families that also are using other communication strategies. They could be using spoken language, but they may also be using cued speech or sign language in addition or as an alternative. All right, so as I noted, there are a number of causes of auditory neuropathy, and one of these is genetic, and the most common genetic form of auditory neuropathy is actually deficiency of a protein called Otoferlin. And let me take a moment to show you how that works. So Otoferlin is a calcium sensor that's expressed in the inner hair cells. So remember, your inner hair cells is the primary transmitter of information from the ear. If you look at the base of the inner hair cells, they have these highly specialized synapses where they're going to connect to the auditory nerve. So these are ribbon synapses, and they're really designed for high rate transmission of information. So your ear can code on the order of 1 to 2,000 hertz pretty faithfully in terms of stimulation, which is remarkable when you compare it to, for instance, like what you can code by tapping your skin with those types of receptors from a frequency perspective. And part of this specialization is Otoferlin. So Otoferlin's involved in this rapid synaptic transmission. It helps tether synaptic vesicles to the presynaptic membrane. It also helps to recycle those vesicles. So in short, it's really involved in this neurotransmission, the communication of information from the inner hair cell to the auditory nerve. The short story is that if a child doesn't have functional Otoferlin protein being produced in their inner hair cell, then this synaptic transmission is disrupted. And that's what causes the child's hearing loss. So what we know is that, and we've learned this from some studies that clinicians have done on the natural history in patients with auditory neuropathy due to Otoferlin deficiency, is that currently most of these kids in this study were being managed with bilateral cochlear implants, and these implants were being placed very early in life. You can see that things have changed over the last 3 decades. This isn't really anything special about Otoferlin deficiency. In fact, just in general, for kids with severe to profound hearing loss, if we go back to the 90s, most of the time we were either putting in a unilateral cochlear implant, or some of the kids were not using cochlear implants. They might have been using hearing aids or some other form of communication. Very few were getting bilateral, so each ear both getting a cochlear implant. But what we've seen over the last couple of decades is that bilateral cochlear implants very early in life have become standard of care for kids with severe to profound hearing loss. Now, how do you get this type of genetic diagnosis? In this case, I'm showing you some very specific information about management strategies for auditory neuropathy that is specifically associated with Otoferlin deficiency. Well, there's been a lot of great work from academic institutions over the last couple of decades who have worked to develop comprehensive hearing loss gene panels. I used to be at Cincinnati Children's Hospital many years ago, and there's a panel there that we ran and is still being run now called Otoseq. But others, the University of Iowa has done a lot of great work on this. Partners, Miami has a really nice panel. They really focused on developing comprehensive testing so that for a child with hearing loss, you could send in a sample and get sequencing. Most of these are NGS panels, and then, get an understanding of whether or not they might have a variants or mutations in any of, in some of the cases, a couple hundred genes that are associated with hearing loss. Now, something that's happened more recently over really the last few years is that we're starting to see now also commercial genetic testing companies who have started offering comprehensive hearing loss panels. And this is really exciting because, the throughput of these companies is obviously a lot higher than we can handle in many academic cores. And so when you start thinking about accessibility and pricing and these types of things, then this gets really exciting about being able to get to molecular diagnoses for many children. But here's what things look like today. As I started this presentation talking about, we've really made massive progress on being able to identify children very early in life using universal newborn hearing screening. So as I mentioned before, from work with people like Carl White, we are seeing now that about 98% of kids are getting tested before they leave the hospital. Now, I will point out one challenge associated specifically with the patients I talked about today who have auditory neuropathy. Some hospitals, when they're doing their newborn hearing screening, they may actually only use the otoacoustic emissions as their screener. And so what I showed you before is that there are kids who have profound hearing loss who have relatively normal outer hair cell responses or otoacoustic emissions. So those kids get missed in screening, and then, of course, it takes longer for them to get into the clinic and get a diagnosis because you have to wait till the family really notices that they're falling behind. The other challenge here is that despite the fact that we're starting to have better and better accessibility to comprehensive hearing loss gene panels, a recent study, this was done by a group associated with CDC and shared at EDDI this year, suggested that it looks like we're only getting genetic testing in about 15% of kids with hearing loss. So what that means is that in most cases, we're not moving to a molecular diagnosis in these kids. We're giving them a diagnosis overall of hearing loss, but we're not actually making the diagnosis of the underlying protein deficiency, despite the fact that we know that most of these kids have a genetic protein deficiency as the cause of their hearing loss. So when folks have talked with parents about genetic testing, this was some nice work done by the group at Miami, and they shared this also this year at the EDDI meeting. They did a little study at their site, which their site is sort of an example of a center that's doing a lot of genetic testing. So in this sample, they actually had half of the parents who had gotten genetic testing for their child, and then the other half who didn't. When they talked to the parents who did not get genetic testing for their child, they asked, why? And in most cases, this had to do with being unaware or the testing never being offered. So there's obviously some other issues here, time, testing, cost, lack of interest. But a big challenge is really just not having the conversation with families or the families being unaware that this is an option. When discussed in this research study, I think families were quite interested in being able to get a genetic diagnosis, and we're happy to have been offered it. Now, my conversations with clinicians have sort of focused around a couple of places where they've seen a lot of value in getting genetic diagnosis for their patients. So one is, as I sort of led with in the beginning, for many of these families, coming to terms with their child's hearing loss is a real challenge. They don't know what to expect. You've shared with them some squiggly lines that suggest that their baby is having difficulty hearing. But it can be difficult to actually accept that diagnosis, particularly if maybe your baby does respond to certain sounds and just not other sounds, right? And so I think the feedback I've gotten from clinicians is that one of the biggest values here is really helping the family come to terms with the diagnosis and start planning with them around management strategies very early in the process. So addition, sort of related to this issue or question about thinking about management strategies, I think an emerging area of research which has been quite interesting, and this comes from Elliot Shearer, who's he did this work at Iowa, but he's now he's a clinician scientist at Boston Children's Hospital. Elliot, in this study, he was looking at outcomes with cochlear implants, and the outcomes in this case are speech perception abilities. And what he asked was, if we do sequencing on all these patients, these are actually adults at this point, we do sequencing, can we understand if the underlying genetic diagnosis had any correlation with their outcomes in terms of how, in terms of what level of speech understanding ability they were able to achieve using an assistive device, in this case, a cochlear implant. And the hypothesis sort of went like this, cochlear implants work by stimulating the auditory nerve. So the idea was, well, perhaps in cases where a genetic diagnosis is able to help us understand the specific structures that are involved in the patient's hearing loss. So in this case, they could be, he's pointed out Otoferlin here, you see. If you say, oh, they have a deficiency of the Otoferlin protein, so what I know is that the cells that are affected here are the inner hair cells, or one of these others that maybe affects the inner and the outer hair cells, something like that. If this is the outcome, since the cochlear implant is stimulating the auditory nerve, one prediction is the patients might do better with the cochlear implant. By contrast, the idea was, because the cochlear implant stimulates the auditory nerve, if you have a genetic protein deficiency that affects the auditory nerve and perhaps its survival and function, then it might provide an outcome that might be less favorable as compared to these patients here. And that's in fact what he had seen within this data set, which these are sort of early data, but what they suggest is that in addition to sort of helping families come to term with genetic diagnosis, helping them sort of get on a path to a management strategy, there is potentially some predictive power in the underlying genetic diagnosis, even if you're using a management approach like an assistive device for the patient. Okay, so in addition to sort of thinking about improving and increasing awareness around genetic diagnosis for children who have already been diagnosed with hearing loss, other ideas that have been discussed by experts in this field recently has been to really question, is it time to start thinking about including genetics in the newborn screening for hearing loss? So right now, the newborn hearing screening is completely done through physiologic and electroacoustic tests in the US. There are other countries that have included some amount of genetic testing for known hearing loss mutations into the process of the newborn screening. But that's not currently part of the process in the US. But this has been an active area that's beginning to be discussed, is what are the benefits here of being able to identify these children even earlier, get to a definitive diagnosis even earlier, and particularly in cases where children might get missed by a standard newborn screening where they may have progressive disease, for instance, get into intervention quite early in the process. We know that in these kids who are born with hearing loss, the earlier the intervention, the better the outcomes in every case. So the earlier we go, we keep just seeing over time that we're getting better and better at delivering outcomes for these kids. through our early screening efforts and early management efforts. The other thing that's happening right now, because of this focus on understanding the underlying genetics of hearing loss, we're now sort of moving from what I would call like a time period in my field of really describing hearing loss geographically. So literally our IC codes are like, you have bilateral sensory neural hearing loss. And then we maybe say, what is the severity of that hearing loss? We're moving from a phase of just describing that overall geography to being able to describe the hearing loss at a molecular level. So we're able to say, not only do you have sensory neural hearing loss, but we know that you're deficient in this specific protein in this specific cell type. And when you get to that level of diagnosis, it catalyzes new ideas about what types of therapeutics could be possible for these patients. And so what you're seeing now are a number of biotechnology companies who are developing AAV-based gene therapies for kids who have congenital hearing loss due to different protein deficiencies. So the one that is expected to go into clinical trials first is this one for Otoferlin deficiency. So you see there are 3 companies here, including the one that I work for, who are developing gene therapies, gene transfer approaches, essentially using AAV to try to start producing Otoferlin protein within these inner hair cells. And the concept is that if you were able to do that, you could potentially instate hearing in a child who was born without hearing. There are others as well. As I mentioned to you before, I mentioned GJB2 earlier. There are several companies who are developing programs for GJB2 for different Usher subtypes, and there's a couple of other genes here mentioned as well. This is an exciting time period because really this level of diagnosis very early in life is really catalyzing opportunities for, development of new therapeutics. So I'm quite excited about the next few years and seeing some of these programs start moving into translation and clinical trials. It's been really a pleasure to be with you all today. As I think I've noted in a number of these slides, newborn hearing screening has been revolutionary for the field of audiology and particularly for these kids who are born with hearing loss. The outcomes that we see now with kids are just night and day compared to a couple of decades ago. And I think what's exciting is there's still room here that we can continue to do even better. And as we think about incorporating new diagnostics, so as time goes on, we get better and better with our diagnostic tools, you can start actually understanding sort of the patient more in terms of what might be causing their hearing loss. And importantly, for me and my colleagues who are doing work on therapeutics, this is really what informs us and directs us on how to develop new therapeutics for these patients. So it's really a pleasure to be with you all today, and I'm quite excited to take any questions and chat about this here at the end. This is my email address. You can feel free to reach out to me if you have any other questions.